Hello everyone, in this video, I'd like to share with you the experiment about bacteria transformation process. Let's go through some background information first. What are bacteria? Bacteria are prokaryotic cells that play an important role in human disease and health. They can cause disease but are also part of the human microbiota and live on our skin, body, and on everyday objects in our environment. When compared to eukaryotic cells, the structure of bacteria is less complex due to the lack of nucleus and membrane-bound organelles such as mitochondria, endoplasmic reticulum, and Golgi complexes. The DH5A competent cells used for today's lab is a kind of bacterial cells which can absorb foreign DNAs. What is a plasmid? Plasmids are small circular pieces of DNA that replicate independently from the host chromosomal DNA. Compared to the billions of millions of bases that make up the entire genome, plasmids typically contain a couple of thousands at most. They are relatively small, stable, and easy to manipulate. In nature, plasmids are usually found in microbes like bacteria. They often contain genes that provide a competitive advantages, giving their host cell an ability that they didn't have before, such as antibiotic resistance. Plasmids are not only useful to bacteria, but they also made themselves indispensable for lab experiments. All plasmids contain an origin of replication. It tells the plasmid where to begin replication. Plasmids often contain genes that are advantageous for survival. One of the most common naturally occurring type of genes is antibiotic resistance, such as ampicillin-resistant gene. These antibiotic-resistant genes help scientists separate out cells that contain plasmids from those they don't. Many plasmids are designed so that scientists can insert genes that they want to be expressed in organisms. The promoter site, which is upstream of the inserted gene on the plasmid, acts as a green light that allows gene transcription. The plasmid used for today's lab is the PUC19 plasmid, which contains the AMP gene and the LAC-Z gene. The AMP gene is ampicillin-resistant gene, which allows the bacteria to grow in the ampicillin-existing environment. Other bacteria, which do not contain the AMP gene, cannot grow in this environment because ampicillin antibiotic can inhibit the synthesis of cell walls. The LAC-Z gene can encode the beta-galactosidase enzyme, which can hydrolyze X-gal, a soluble colorless compound consisting of galactose linked to a substitute indole, to form a blue product, which is easier to be observed. But LAC-Z gene transcription only happens in the presence of galactose. In this experiment, IPTG, a non-metabolizable analog of galactose, was used to induce the expression of the LAC-Z gene. Let's start our experiment. Label one microcentrifuge tube plus DNA and a second microcentrifuge tube minus DNA. Label four agar plates which contain different elements as the picture shows. Transfer 50 microliter of competent cells to each of the tubes. Add 10 microliter of the PUC plasmid solution to the tube labeled with a plus sign. Do not add the PUC plasmid to the tube labeled with a minus sign. Incubate both tubes on ice for 20 minutes. Carry the ice container over to the hot water bath, which is set at 42 degrees. Insert both tubes into a float and immediately place the float into the warm water for exactly 45 seconds. After 45 seconds heat shock, return the tubes on the ice container and incubate them for 2 minutes. Pipette 500 microliter of LB medium into each tube. Gently mix the tubes by tapping them with fingers. Incubate both tubes for 20 minutes in an incubator set at 37 degrees and 200 RMP. After 20 minutes incubation, spin the tubes down for 60 seconds in a microcentrifuge. Remove 300 microliter of the supernatant. Resuspend the cell pellets in the remaining 250 microliter of liquid by pipetting gently up and down. Transfer 100 microliter of the plus plasmid and the minus plasmid solution onto each of the plates correspondingly. Use 6 to 10 clean sterile glass beads for each plate to spread the cell solution by swirling the plate around. Stack the plates and tap them together. Place the stack of plates upside down in a 37-degree incubator until the next day. Then, the next day, we are able to see blue colonies on the plate which contain the PUC19 plasmid, LB, and AMP. No visible colonies appear on the plate which contain LB and AMP without the PUC19 plasmid. Using a microscope, white and blue colonies can be found on the plate which contain the PUC19 plasmid and LB. Using a microscope, white colonies can be found on the plate which contain LB without the PUC19 plasmid. This experiment illustrated that bacterial cells could complete the transformation process by picking up the foreign DNA plasmid under some circumstances. So, how does this relate to our lives? Because of the versatility the plasmid colony process have, there is a whole area of genes that scientists can introduce into the cell. For example, if you want to track a specific species of bacteria in your population, you can insert a GFP-expressing gene into your plasmid and transform it into your bacteria. Your cells will fluoresce, making them easier to be found. Or you are studying the effect of specific protein on phenotype. You can insert a gene of interest into the plasmid, moving it into your cell, and look for changes in the cell. Furthermore, plasmid DNA technology can cure DNA defect diseases which cannot be cured by traditional treatment. Scientists can insert a correct version of a broken gene into a plasmid and place it inside bacteria. Then, using bacteria's natural rapid reproduction, enough copies of DNA for patient treatment or research are produced. Using some viruses or AAV, the plasmid containing transgenes are delivered inside infected cells and allow them to create the needed protein, delivering treatment to the patients. Plasmid DNA technology can also be used for producing vaccine. After delivering the plasmid which contain the virus DNA into the bacteria, we can gain the virus code protein and use them in vaccination. Plasmid are also useful in many other fields, like the gene engineering. These are all about the experiment, and thank you for watching.